Hey guys, I'm Sandhya and today we are here in the Science Maths course and today we are going to talk about microscope and I hope you people are aware of microscopy. So microscopy is a science where actually we are dealing with the specimens and we are investigating it under a microscope and we are having different types of microscope. In this video today we will be talking about a very common microscope which is present in every biological lab and I am talking about dissecting microscope, also called as stereo or stereoscope. Microscope which I am talking about. By the way, we are having multiple types of dissecting microscope, but this is a very common one which you can easily get in any store, even online as well. So let's talk about this microscope. We use it for very lower magnification. It's an optical microscope. Now let me just show you by using a specific specimen that how are we going to investigate it under this lens. So here, you know what happens in this? So there is a reflection of light from the surface of object rather than the transmission part. Only the light is being reflected by whatever object you are keeping over here. I'll first of all tell you that what are the parts of this microscope. This is the eyepiece and in the eyepiece we are having this lens. Let me first of all tell you that the magnification of the lens is quite lower. Here I am having just 10x. It goes from 10x to 20 till 100x as well, but here this is a very low magnification power that I am having over here. It is just 10x because the best thing about this microscope is this is gonna provide you a larger working distance. How come? Here you can see the focus, right? This is the focus part. It's a focus knob present over here. Because of this you can see that this much is the area for my working area, which is not possible in the compound microscope. That's the best part about this. Another important thing is that this can give you a three-dimensional view. I'll show you something very easy that the objects you can see here is you cannot see this ring in a compound microscope because this is gonna make it opaque. You can watch any kind of like flowers, maybe a rock or maybe coins as well. So these are the opaque objects. So the light is not passing through them. So that's the easiest path to see it. It works similar to the magnifying glass as well. So here there something is written on the ring. If I try to show you in the microscope, first of all it is important whether the light is passing or not. If I just try to show you, you'll be able to see this. See. First of all, it's giving me a larger working distance, and if I try to show you, see this is looking so minute, right? Yeah, but if I'll just increase it, just look at the letters now. They are easily getting magnified. Let us focus the light. Yeah, yes. Yeah, the letters are easily getting magnified. Just make sure that you are working on light in a better way. See. And this is so small, but here inside it is getting magnified. So this is, by the way, a ring. While talking about a biology, I'll just show you with the help of a flower that I'm having here today with me. I'm having a hibiscus flower. When I show you the flower, you can see we are having different parts of flower. I'm having some forceps and needles. Yeah, so I'll just remove the sepals and the petals from here. So we are having, this is a bisexual flower that you can see. We are having the stigma, then we are having the style, then we are having the ovary. And here we are having the androecium, that the stamen part is present. I'll just take out one stamen from this forceps. So this is the stamen part and I'll keep it on the stage. This is the stage plate. We're going to keep the stamens. You need multiple tools while you're performing the bio experiments. Please go through my dissecting box. Link I'll be giving in the description box. Where you'll be able to see different types of tools that has to be there while working in the biology lab, and you can easily get them in the dissection box. So here is my, so this is my stamen where I'm having anther and filament both. So now I'll be trying to magnify this first of all, with the help of this microscope. So can you see this? It's looking so small right now. Yeah. So now as I try to focus it, you can see this is gonna increase in size. This is how you can magnify it. Even if I try to just observe the small anther, if I separate it with the help of the needle, see, can you see this? I'm just trying to work on this, and this is gonna ye kaam you cannot do it in the compound microscope, because here I'm easily working on the stage. This is giving me a working distance. In a 3D process, I'll be able to work with any specimen that I'm gonna keep here. You can see this anther and the stamen. But if you see it with your naked eye, this is going to be very minute and difficult for you to observe it. So that's the beauty of this dissecting microscope. This is, I'm just showing you with the help of a pollen. I'm just trying to show you the stamen part. But you can use any specimen, and this is a very common microscope. But very soon I'll be also letting you know about the compound microscope that is going to magnify the image at a very higher level. So we'll be talking about the compound microscope in our next video. I hope you like this. Please go like and subscribe the same.